Thank you very much, Dr. Zackular. That was a wonderful presentation. Yeah, really appreciate the kind of ability for your research to cross these different scales, going from like modeling to experiment to validate the mechanisms. Yeah, we actually have quite a few questions here for you. So I'm going to go in order of those voted. So first one says, on one slide, I think it was one of your first slides, looking at the associations with commensals and C. diff, someone noted that there seemed to be another commensal previously associated with susceptibility. They asked, have you looked beyond the enterococci?
Yeah, so there is quite a few organisms that are positively associated with C. diff infection, and by no means are the enterococci the only ones that we think are important and that we're interested in. And I think in the Buffy et al. Study, there was a few that popped up through the study. They did a couple different ways of looking at this, and one was the lactobacilli. We tried to look at the lactobacilli, one because they also use the arginine deaminase system. But the problem with the lactobacilli is that they actually produce a lot of factors that inhibit C. diff as well. And so, Rob Britton's done quite a bit of work on this, that, you kind of have to find the right strain that isn't inhibiting C. diff and another mechanism to see if it can positively associate it with C. diff in another way. But it kind of goes towards context dependency, where the enterococci seem to really do well with C. diff. They seem to be positively associated in, a lot of studies. And then of course in our mice, and then when we're studying them and trying to understand how they interact, they partition resources really well. They both, are positively associated with the same, perturbations in the gut. So they see each other quite often. And I think that they're easy to kind of study in this context, but others like the lactobacilli, it might be very strain specific. It might be biogeographically specific, thinking about where they might see each other. And so we're very interested in these kind of nuanced looks. And then of course you might ask yourself, like, I'm just filibustering until we, make sure that
No, you're doing great. We also have a bunch of questions. So
yeah. I just think it's like a really cool way to think about it. And we're only looking at 2 organisms here. And if you go back to that slide I showed, we have a lot of patients that have no enterococcus during infection. So what's going on with them, right? They could be just as severe. And so we're very interested in all of that.
Yeah, that's really interesting what you mentioned about the lactobacillus too, because as I think one of our other speakers noted, those are traditionally thought of as beneficial commensals, but there's spectrum, and they're not all good guys, and context matters.
It's all about context.
Okay, yeah, so moving right along, next question. Yeah, I think in the very beginning you showed the mouse models and how you showed that this antibiotic treatment that could potentially knock out your enterococci was like some of your first results showing how they were associated with C. diff infection. The question is, in your antibiotic treatment, like, how did the treatment affect the other commensals, and were the other commensals present?
Yeah, absolutely. And so those antibiotic treatments aren't like, they're depleting the microbiome in terms of biomass, and cefepime alone even really alters the microbiota. And then the cocktail of cef and vanc is really dramatically perturbing the community. We haven't looked at what other organisms are, like negatively, like, what the vancomycin might be knocking out that also could be associated. We were taking a sort of enterococcus specific or enterococcus centric look at this, mostly because the enterococcus are so clinically relevant to C. diff infection. But we've started to go back and like look at the same system and think more broadly about the community and do metagenomic sequencing and trying to understand which other organisms are associated with this, and then which metabolites are associated with this. Look, why exactly are certain antibiotic cocktails more or less conducive to C. diff colonization early on? And is this, kind of life beyond just bile acids? We think a lot about bile acids in C. diff infection because they're so important. But we think that this is more complicated than bile acids and likely other nutrients and yeah, different factors. And we certainly think that the enterococci are providing fermentable amino acids, but there, what else could be going on there? What other organisms might be positively or negatively associated in those antibiotic treatments? We're very interested in.
That sounds great. Okay, so next up, participant asked, could you please expand on the horizontal gene transfer result that you touched upon in the talk?
Yeah, absolutely. So we what we were able to do is take, because we see so often co-infections or co-colonization of VRE in the gut during C. diff infection, we were able to do analyses with Derry Van Tyne's group at the University of Pittsburgh from a cohort of patients at Pitt that had both of these organisms and look for putative metabolic or putative mobile genetic element transfer. And what we find is really, really robust shuttling of antibiotic resistance genes specifically. The most prominent of those is TN916, which is a well-known transposon. We actually use it in some strains to get DNA into C. diff, and it encodes its own conjugative element on it and has tetracycline resistance. And so we can actually actively see that moving between the two, during co-infections. So a lot of the things that we found were clusters of, mobile genetic elements associated with antibiotic resistance. However, one little bit of the story that was really, really fascinating that we're very interested in is we actually find that there are genes that go from, enterococcus love to share DNA. They love to move around plasmids and transposons. But we actually found an example of a repeated transfer of a C. diff collagen binding gene to VRE. And what was interesting is that this was a sorted collagen binding or predicted collagen binding protein from C. diff that we were finding in VRE readily. And if you take it and you clone it into the lab strain that doesn't have this, it binds collagen better. And so it can sort this gene from C. diff, put it on its surface and bind collagen better and make better biofilms. And so it was actually really, really cool because they're finding each other a lot in the gut because of this antibiotic treatment and the perturbation that we're giving. They grow to really high levels. And then it seems like it's not just metabolic crosstalk, but it's DNA crosstalk. And in some cases, we're finding examples of functional proteins moving between the two that might change the niche of each of these. And context, again, is, really important there, right? So. Yeah,
that's fascinating. Yeah, do you think, just to kind of follow up on that, you think then that would suggest either potential or maybe some past history of coevolution of these two?
Yeah, so this is it's really interesting to think about, so there's no doubt that these two organisms see each other often in the healthcare setting. And there's the examples of VRE faecium evolution, and specialization to the healthcare settings, right? The VRE faecium that are causing infections are really, really divergent from, regular faecium. They're very, very well suited for the hospital environment. And I find that really fascinating. So there's clearly a lot of action that can happen in the short period of time that we've been using antibiotics, that these two see each other and maybe have co-evolved in that case. But where I think this could be more of a long-term, coevolution that might just manifest more recently is that these are two organisms that colonize infants very, very readily. And so enterococci are among the first organisms that colonize a baby during succession of the microbiome. And we find C. diff in, most babies. And the question is that a new thing from, hygiene hypothesis? Has C. diff always been in babies? It would kind of be cool to think about that. But maybe they've been co-evolving kind of early in succession of the microbiome when it's not overt disease, long before we then kind of pulled it out with antibiotic treatment. But I'm kind of hand waving there. But that's kind of my answer to that.
Yeah, that's really interesting. Also, yeah, you mentioned, and this is a fact I'm familiar with too, the fact that, yeah, lots of infants tend to be colonized by C. diff. Yeah, why do you think that is? Do you think that's something that's always been true? Is it maybe a more modern phenomena? Like, is it coming from the hospital or the mother?
Yeah. So we're studying this very actively. It's so weird. It's so cool and weird, right? Like these weird phenomena that everyone knows, but you can't explain. So the answer is we don't really know. But the antibiotic treated gut looks a lot like an infant gut, right? Like in terms of bile acids, in terms of the lack of diversity, the simplicity, you haven't grown out. And the thing that kind of saves you, or at least we think saves you, is antibodies and breastfeeding from mom and things like that. But I suspect it's the antibiotic gut. The antibiotic gut is a lot like the infant gut, and C. diff is just taking advantage of open niches and is a spore former, so it's kind of around, right? And so that's kind of why I think it probably is colonizing infants so readily, because it's open niches and there's lots of primary bile acids, and it just kind of goes. But why it's acting the way it does in infants, I don't think anyone knows. Why, like, infants don't get sick from C. diff, which is, I have a 3-day-old here, they get sick from everything, right? Like, so like, why don't they get sick from C. diff? It's a mystery that's very cool.
Yeah, that's fascinating.
Yeah.
Maybe one more question, because I looked and it looks like Affiliate was able to make it.
Oh, awesome.
So yeah, last question. I think this connects back to one of the previous talks. So you had mentioned this phenomena with arginine and the ability of the enterococci to metabolize it and produce ornithine for C. diff, if I got that correctly.
That's right.
So the question is, do you think an organism like Elenta maybe could be used to outcompete this enterococci and ablate maybe its benefit to C. diff if it's consuming the arginine?
Yeah, it's a really good question. And I think what actually is happening with Elenta is that I would assume it's playing the same role as enterococcus, right? Because it's using arginine and making ornithine, and C. diff responds to that in a more pathogenic way. I think what we would want to do is try to identify ways to kind of break, like to maybe compete with the ornithine, right? Like maybe you could, like thinking about like breaking these arrows, right? Like these context dependent interactions that are driving changes in C. diff fitness. Could you compete with these different arrows in terms of, or, alter that communication? The way that, we would think about it is that you could certainly try to compete for the arginine, but I think it might have the same effect on C. diff. We're not sure. Maybe the way to think about it is that could you inhibit the arginine deamination, or could you compete for the ornithine? Like these are the ways that we're thinking about it. But actually, as I talk about it, it's a really good question. I think, like, how could you ecologically break these chains is exactly what we're thinking about, because our study was able to show that by altering the metabolism of another organism that isn't the pathogen, you can change the pathogen, the outcome of the infection, and the pathogen's behavior. And so that gets to start, you start to think about what that means. It's a cool proof of principle that targeting bystanders can change the entire chain. And so, like, creative ways to think about that is, I think, what you all out there will be solving in the future for sure.
That's great. Well, thank you so much.
Thank you, Alex.
And for all these insights. Yeah.